Hello, Mr. Lambden. Today I'm going to be making you my video about DNA replication, and I decided to utilize paper and markers because I had lots of colored markers laying around with my sister. Also, I apologize if you hear my family at some point in this video because they are singing Disney songs really loud downstairs, so I apologize if you hear them throughout the course of this video. To begin with DNA replication, helicase, which I have as a purple triangle, comes through and splits all the hydrogen bonds between the nitrogen bases, making them split like this. And then we have gyrase, which kind of makes sure that the two DNA that are still intertwined, the two sides of DNA that are still intertwined on this side, that they don't unwind additionally from helicase. And then I have these really small SSB, or single-sided binding proteins on the side, which makes sure that the now split DNA sides stay in their place during replication. I'm going to take these away so that we can focus on one half of DNA at a time, because that's how I organize it. And I'm going to be demonstrating the leading strand first, which means that there's really no Okazaki fragments and that it is one continuous flow of building. So to begin, we have primase, which makes an RNA primer so that the DNA polymerase III knows where to go to build DNA. And so, since this is in the 3 to 5 direction, the RNA primer will be building, or the new DNA molecule will be building in the 5 to 3 direction. So, this should be 5. So now this is my RNA primer, considering that it has uracil instead of thymine. And then DNA polymerase III comes in and starts building DNA off of the primer and builds a replicate of the other side. Once this is made, DNA polymerase I comes in and replaces the RNA primer with DNA, so I change the uracil back to thymine. And then we have ligase, which I can find right here, that kind of glues the segments together. This is one continuous cycle, and it doesn't backtrack like the lagging strand does, which I will now demonstrate. Okay. So I have lagging strand. Okay, so in a similar way, primase starts off by making an RNA primer. And actually, I'm going to start on this side. Because it goes in the 5 to 3 direction. So primase starts and makes an RNA, which is this portion right here. And it makes an RNA primer, which I have represented by purple dots on the C. And then DNA polymerase III makes this part of DNA. Since this is going in the 5 to 3 part on the lagging strand, it kind of jumps back, considering that they open in opposite directions. So, due to this, primase once again needs to be involved here and make another RNA primer, which I have made with the purple dots. And then DNA polymerase III once again makes 2 new little nucleotides of DNA. Similar to the leading strand, if I can find it, DNA polymerase III, or DNA polymerase I, then replaces the RNA with DNA, which I have there. And then, once again, ligase glues the Okazaki fragments together because these are Okazaki fragments with the RNA primer and the DNA chunk. I think that's it. So then you would have, if I can get it all in the same, I don't know if that's all the same, but so then I have 2 of DNA strands, and then I have the parent strand here and the parent strand here. So yeah, I hope you enjoyed this. I'm sorry it's messy and I gibbered up my words sometimes, but there you go. Have a good day, Mr. Lambden. Bye.